Deep within almost every cell of the human body sit specialized bioenergetic structures known as mitochondria. These double-membraned organelles act as microscopic power generators, converting oxygen and nutritional substrate molecules into adenosine triphosphate, the universal currency of cellular energy. In peak biological youth, healthy mitochondria continuously produce energetic ADP while efficiently managing byproduct free radicals and maintaining internal structural integrity. However, as decades of biological life elapse, oxidative stress, metabolic strain, and environmental exposures cause cumulative structural damage to mitochondrial DNA, membrane proteins, and internal cristae. Instead of cleanly synthesizing cellular energy, failing mitochondria begin to leak high levels of reactive oxygen species into the surrounding cytoplasm. This biochemical leakage damages surrounding cellular architecture, impairs protein synthesis, and triggers chronic low-grade systemic inflammation throughout surrounding tissue beds. Systemic mitochondrial decay is now recognized across modern biological literature as a primary hallmark of human aging, driving age-related sarcopenia, metabolic slowdown, and chronic fatigue. For decades, classical biology viewed this progressive mitochondrial deterioration as an inevitable, irreversible downward trajectory of human biology. However, modern cellular physiology has revealed that the body possesses a highly sophisticated internal quality control mechanism dedicated to organelle renewal, a process termed mitophagy. Mitophagy is a specialized form of selective autophagy designed specifically to isolate, dismantle, and recycle damaged or inefficient mitochondria inside living cells. During optimal mitophagy, molecular sensors scan mitochondrial outer membranes, identifying damaged organelles that have lost their electrochemical membrane potential. Once tagged, these compromised powerhouses are enveloped by double-membrane autophagosomes and fused with lysosomal digestive vesicles, breaking down organelle structures into basic amino acids, fatty acids, and mineral building blocks. These recycled cellular raw materials are then repurposed to synthesize new, highly efficient mitochondria through a complementary biological process called mitochondrial biogenesis. When mitophagy operates cleanly, cells continuously purge compromised power generators, maintaining high metabolic capacity, metabolic flexibility, and low baseline oxidative stress. The critical vulnerability in aging human physiology is that key youth-associated signaling pathways required to initiate mitophagy steadily decline over time. As signaling proteins like AMPK and sirtuin-1 lose sensitivity, damaged mitochondria escape detection and linger inside skeletal muscle fibers, neural networks, and cardiac tissue. This cellular congestion starves cells of energy while generating constant inflammatory signaling that accelerates biological aging across organ systems. In recent years, longevity researchers and cellular biologists discovered a unique postbiotic molecule capable of directly stimulating this dormant mitophagy machinery, Urolithin A. Unlike synthetic pharmaceuticals engineered in industrial laboratories, Urolithin A is a natural metabolite produced through complex biochemical interactions between plant polyphenol and the human gut microbiome. When you consume dietary sources rich in elagitannins and ellagic acid, such as pomegranates, walnuts, pecans, and wild berries, large complex polyphenol molecules enter the gastrointestinal tract. Because elagitannins are too large to be directly absorbed through the intestinal epithelium into the bloodstream, they travel down into the colon. Inside the large intestine, specialized commensal gut microflora ferment these dietary precursors, performing sequential enzymatic cleavages that ultimately yield smaller, highly bioavailable urolithin compounds. Among these metabolites, Urolithin A possesses superior lipophilic properties, enabling it to cross the intestinal mucosal barrier, enter systemic circulation, and penetrate target cell membranes throughout the body. Once inside target cells, Urolithin A upregulates key regulatory proteins responsible for tagging dysfunctional mitochondria, notably Parkin and PINK1. These molecular markers attach ubiquitin chains to damaged outer membranes, signaling the cell's autophagic machinery to initiate immediate lysosomal engulfment and breakdown. By promoting the selective removal of degraded organelles, urolithin A clears cellular congestion, allowing remaining healthy mitochondria to expand and replicate through fission and biogenesis. This continuous cellular renewal enhances total ADP production, restores metabolic efficiency, and lowers baseline cellular oxidative stress across targeted tissues. However, nutritional scientists quickly discovered a major biological challenge regarding natural urolithin A production across human populations. Clinical analysis reveals that despite eating identical quantities of pomegranate or walnut precursors, only an estimated 12 to 40% of adults actually possess the specific gut microbiome strains needed to synthesize urolithin A. The primary bacterial species responsible for this complex biotransformation belong to specialized lineages within the Clostridioides order, including Gordonobacter and Elagibacter species. If your gut microbiome lacks these specific bacterial populations due to historical antibiotic usage, ultra-processed dietary patterns, or environmental factors, eating pomegranates will yield minimal to zero urolithin A in your bloodstream. This significant microbial variation explains why dietary interventions often produce wildly inconsistent health outcomes across different human subjects. To overcome this microbial bottleneck, researchers began administering pure, direct urolithin A in controlled human clinical trials. In landmark clinical studies published in peer-reviewed scientific journals, middle-aged and elderly human subjects received daily oral urolithin A supplementation over multiple months. The clinical findings demonstrated significant improvements in skeletal muscle endurance, increased cellular ADP generation, and measurable declines in circulating inflammatory markers like C-reactive protein. Analysis of skeletal muscle tissue samples confirmed a marked upregulation of core mitophagy gene expression alongside increased density of healthy mitochondrial cristae within muscle fibers. Subject groups receiving urolithin A achieved improved physical walking distances and enhanced muscle strength without alterations to their baseline physical exercise programs. Beyond skeletal muscle architecture, neuroscientists are actively investigating the neuroprotective impacts of urolithin A within the human central nervous system. Brain tissue is extraordinarily energy demanding, consuming approximately 20% of total resting body energy despite representing only 2% of total body weight. Neurons rely almost entirely on efficient mitochondrial energy output to maintain membrane polarization, propel neurotransmitter synthesis, and drive continuous synaptic signaling across neural circuits. When mitochondrial turnover slows within neurons, the accumulation of broken organelles triggers localized neuroinflammation and impairs cognitive processing speed. Preclinical models demonstrate that urolithin A successfully crosses the blood-brain barrier, stimulating neuronal mitophagy and protecting delicate hippocampal structures from oxidative degradation. Cardiovascular researchers have similarly observed protective effects within vascular endothelial tissue, where dense mitochondrial networks are vital for synthesizing nitric oxide and maintaining arterial flexibility. While direct urolithin A supplementation offers a targeted intervention, supporting natural microbial production remains an essential foundation of long-term metabolic health. Consuming a diverse spectrum of polyphenol-dense whole foods provides the necessary biochemical precursors required by beneficial gut microflora. Fresh pomegranate arils, raw walnuts, red raspberries, blackberries, and polyphenol-rich botanical teas offer rich natural concentrations of elagitannins. Pairing these polyphenol precursor foods with high-fiber plant foods promotes a gastrointestinal environment optimized for microbial diversity and postbiotic generation. However, It remains important to recognize that dietary optimization and cellular postbiotics serve as supportive components of overall wellness rather than singular solutions. As with all health science content, this video provides general educational insights and does not constitute individual medical advice. Always consult your qualified medical provider before implementing dietary changes or new supplement protocols. Understanding the cellular mechanisms governing mitochondrial renewal allows you to make informed decisions about your long-term energy, metabolism, and longevity goals. By activating targeted quality control pathways like mitophagy, modern nutritional science provides powerful tools to sustain cellular vitality into every chapter of life. When damaged cellular power generators are systematically recycled and renewed, your body retains the energetic foundation necessary to thrive. Research into Urolithin A represents a transformative step forward from broad antioxidant approaches toward precise, mechanism-based cellular rejuvenation. As ongoing scientific research continues to illuminate gut mitochondrial crosstalk, Urolithin A stands out as a remarkable natural key to unlocking human longevity